Okay, so good afternoon, everybody. It's my pleasure to introduce Bastian Rieck, who's joining us from Helmholtz, Munich, the pioneer campus in the Institute for AI for Health. Bastian is the PI of the IDOS group, which focuses on biomedical applications of AI research. He has previously been at the ETH in Zurich with Carsten Borgward, and before that he did his PhD in the University of Heidelberg. So please, Bastian, take it away.
Thank you very much for having me. It's amazing to be given this opportunity so early in the year to talk about something very cool and something very exciting for me. So this talk will be two talks or two papers rolled into one, or actually a little bit more than that. And I aim to give you a little bit of an overview here. And I opted for a little bit of a let's say action type of title, namely zoom and enhance towards multi-scale representations in the life sciences, with the understanding that there's of course a little bit of a joke in there, but it sometimes is a good idea to zoom in a little bit and try to figure out what is going on beneath the surface. So my first motivation for this is this classical TV show that maybe some of you still remember, and if not, then that pretty much dates me all right. So here's the idea, of course, that you can magnify stuff, but that doesn't necessarily make it any clearer. At least it does not necessarily do that in science. And so we need new methods to work with data on different resolutions and at different levels of such resolutions in order to make sense of them. Because sometimes we might be interested in the bigger thing, and sometimes we might be interested in a specific aspect of that, of our systems. And so it pays off to be capable of looking at data from different angles. Now, as a warm-up, how does that look for real data? Well, oftentimes it's all about finding the right perspective. And when you found the right perspective, you might not like what you see there. So for instance, when you take this graph, and I've deceptively numbered the x-axis here, so maybe some of you can guess what is going on. When you look at this graph here, we will be saying, oh, well, that's a time series, and it's probably like, not changing a lot here. So not a lot of stuff going on, nothing to be worried about, right? But when you zoom out, you see, oh, yikes, something is going on, and we might not like it. Now, this is, of course, a well-known graph from climate change. It's a little bit of a misnomer for me because I'm not going to talk to you about climate change today because I have no idea about that topic, but I like the graph and I like the aspects it symbolizes. Now, what am I going to talk to you about? Well, in the first part, I'm going to talk to you about antimicrobial resistance prediction. And now a big disclaimer, I'm not a doctor. Well, I am, but not the kind who helps people, or maybe only the kind who helps people through proxy, namely through machine learning. So my biological background is, let's say, sketchy. I'm interested in it, obviously, but I also understand that I have to rely on strong main partners to make sense of that. So please, they briefed me and they gave me a lot of instructions, and any errors I make are certainly errors of my own misunderstanding, but do let me know if there's something unclear, and don't wonder too much if I say something that is biologically implausible. This could very well be my own misunderstanding. Now, antimicrobial resistance is a little bit of a problematic topic for a while now. So of course, we all have seen the effects of the ongoing pandemic, and the pandemic is something completely different. This has nothing to do with antimicrobial resistance necessarily, but antimicrobial resistance lurks a little bit behind that pandemic now. And some recent articles are even saying that, well, this is going to be the next big health emergency, or others are even using the term pandemic itself in order to confront this problem of antibiotic resistance. And of course, there's always these reports going around that millions of people are dying from infections that are resistant to drugs in the hospital. And maybe some of you already encountered this. I mean, I hope not, but if you get an infection, then it's sometimes you have to use these broadband antibiotics because it's a little bit unclear what will take and what won't take. And there's even a whole subfield around it. It's quite fascinating from my point of view, where people are discussing good antimicrobial stewardship, because the idea of just using broadband antibiotics for everything is not a good. It's not a good strategy. It's a good strategy to ensure that we will have more resistant strains. So there's clearly an issue here that we address to solve, or that we address to alleviate to some extent. Now, what are we going to do to alleviate this, or what will we try out in the future? Well, one thing that we encountered in our research is this idea of using mass spectrometry in the clinic, and this is almost part of the daily clinical practice, at least in some hospitals. The idea behind that, and again, I have to remain a little bit sketchy because it's not exactly my forte, is so-called MALDI-TOF mass spectrometry. Now, MALDI stands for matrix-assisted laser desorption ionization time of flight mass spectrometry. And I'm glad that I could pronounce that without stumbling. Let me give you a brief overview of what this means. Well, in MALDI-TOF MS, you have a laser source, and the laser source is directed at your mixing sample matrix. So it's your sample, the thing you want to analyze, together with some kind of matrix material. And this is put on a plate. By putting the laser, by focusing the laser on that, you basically superheat the sample, which results in some desorption. So it breaks up, and you gain some molecular ions in the as a result of that. And these molecular ions that occur from this reaction are the things that you then measure with the usual time of flight mass spectrometry. The idea being that there are certain time of flight numbers that are kind of characteristic for certain types of molecular ions. Now, what you can do with this technology, so in my mind, I treat this as a black box because I don't have to use this, but what you gain from MALDI-TOF MS is you obtain a quick overview of the microbial composition of a sample. And interestingly, the spectra that you get out of there, we'll see an example in a minute, they are known to be highly characteristic of a microbial species. And this has the effect of making MALDI-TOF MS a standard tool for the identification of microbial species in clinical practice. So this is what spectrum looks like. It's pretty innocuous, right? You just have your M/Z ratio here, and then you have the measured intensity. You can see that there are some that I cut off this ratio a little bit. So in practice, you might get broader spectra, but this all depends a little bit on the machines that you're using. There are all kinds of interesting parameters by the creators of these machines to be encountered here. And well, this is just an example of how it might look, but this is a real example. So it's not something that I made up. Now, what's the standard workflow when you have an infection that needs to be treated? Well, the standard workflow is the line on the top, essentially. You go to the hospital, they do some sample collection of your, so they collect some samples of your infection, depending on you have to cough on something or whatever. Then these samples are being cultured for up to 24 hours. And standard MALDI-TOF is being used in order to do the preliminary species identification potentially. Then the samples are furthermore cultured for up to 72 hours. And during this culturing process, AMR measurements, so antimicrobial resistance measurements, are being taken. So the way I understand this is that the clinicians or the lab technicians provide some antibiotics to the samples that they took from you, and they just wait for up to 72 hours to see what happens. And then they can decide whether the sample is resistant to a specific antibiotic or whether it's susceptible to that antibiotic. There are also some kind of mixed cases, but we're going to ignore this in this talk because, well, it goes a little bit beyond what I want to talk to you today. But anyway, from these measurements then, the treatment decision is being made by the clinician or by the physician. So based on the results of the long cultured samples and the AMR measurements, you will be prescribed a specific antibiotic, and hopefully this will combat the effects of the infection. Now, what we are proposing, or what we want to do in this project, is we want to use the MALDI-TOF data that is being generated anyway. We want to use that data and do some AMR prediction, some antimicrobial resistance prediction with machine learning algorithms in order to skip these 2 steps here and go directly to the treatment. At least I have to say that's the vision of what we want to do, where we are obviously not quite there yet, but we're doing some progress here. And it's of course clear that why this would be useful, because in the worst case, you will have to wait 3 days for your AMR measurements. So if there is an algorithm that is sufficiently precise and sufficiently accurate in predicting which antibiotic would be susceptible to the infection, or conversely, which one wouldn't, then of course you can save some time, and saving some time for infections always means that the patient well-being is ultimately increased quite a lot, because that means that you're not doing the wrong treatment, and you're doing the right treatment as early as possible. And so that's kind of the goal. That's the overall thing that we want to accomplish. So how are we going to do this? Before we enter into this, let me just say something. I just realized that I can't see any questions or any chats in between, so I'm just looking at my slides. So if you have any questions in the meantime, you also feel free to interrupt me, but we can also, of course, take questions after the talk. So whatever works for you. But if there's something that you don't understand, just tell me in the middle, because that might make it easier for me to continue. And anyway, how is this typically done? How are these spectra being used for machine learning algorithms? There's 2 pre-processing pipelines that I want to briefly talk to you about. The one on the left is let's say the classical one, the established one. The one on the right is one that we tried out in one of our papers. Then going into the details in a few slides. The first, let's say the classical pipeline uses a very cool package, a very cool R package called MALDI-Quant, which is specifically geared towards analyzing mass spectrometry data, and has quite a lot of interesting steps in there which are required. So one is the variance stabilization. So you want to have a better variance of your individual spectrums. There are some smoothing steps which remove some of the problematic outliers, or which remove some of the measurements that might not make sense, because mass spectrometry is of course a fundamentally stochastic process in some sense. Then there's a step called baseline removal, which ensures that you get the right intensities that you would anticipate if you were to do this in as a thought experiment. There's some things like intensity calibration, so that the intensities match what you would expect, and intensity trimming, so that the intensities are not too high or too low. To me, this is similar to gradient clipping, for instance, what you're doing in machine learning. But the bottom line is that this pipeline is very well established, and it works, but it also has a lot of parameter choices. And of course, parameter choices always influence the results to a certain extent, or they make it harder to train a classifier in the end, because you have to walk over them somehow and understand what their implications are. So the first thing that we wanted to do as part of this project is we wanted to figure out whether there are easier ways to deal with spectra in practice. And to this end, we developed another pipeline that just has a single step. Namely, we treat the spectrum as a function, and we use the so-called prominence or persistence of critical points of the function as a proxy for the heights, for the intensities of a peak in the spectrum. Now, this is one example of this zoom and enhance that I wanted to show you, multi-scale view of the data, if you will. And for this, I need a brief digression so that you understand what we're talking about. And here, being based in Munich and in Switzerland, of course, we all know that the mountains are quite nice and very nice for leisure and for skiing and whatnot. And for me, a mountain range is nothing but a very simple one-dimensional function. So I'm just looking at the heights above sea level, for instance. And for this simple one-dimensional function, we can prescribe the so-called critical points. Every one of us knows those. We've learned about these critical points in high school. We just maybe didn't call them that. We just call them maxima or minima, or maybe saddle points. But those are the so-called critical points of a general function. Now, the concept of prominence that we used for dealing with our spectra actually comes from mountaineering. I'm not kidding you. This is actually being done here. And it comes from a very simple observation, namely, when you climb that mountain here, that peak here, it would be right for you to say, oh, well, I climbed a mountain that has height h1, right? But now as you're on top of that mountain, suppose you go to this other mountain here. Well, in that sense, you wouldn't say that this mountain has height h2, of course, over height over the sea level, but it's not quite fair for you to say, well, and then I climbed another mountain of that size, because that's not really the height that you have to climb once you're already at that peak here, right? So in fact, what happens is that mountaineers assign these peaks a so-called prominence value, and this is calculated by looking at the contour lines of the mountains and checking what the maximum height is that you can go in terms of the contours of the peak without meeting a higher peak. And the maximum that you get here is called the prominence of that peak. The idea being that if you have a lot of peaks that ordinarily, if you measure them from sea level, would have more or less similar values, similar heights, it might still not be the case that they're kind of similar in terms of the prominence, because they're just one out of many peaks of the mountain range. And this concept is known as prominence in mountaineering, but in mathematics is actually known as something else. It's known as topological persistence, a concept from topological data analysis. And while I will show another slide about this topological data analysis things as well in a second, it would be remiss for me to assume that I can show you all the cool things that are going on here. It's an emerging topic that deals with analyzing data at multiple scales. And if you're interested in this, feel free to contact me. And for instance, you could read a recent review of ours called A Survey of Topological Machine Learning Methods, in which we outline a few of the recent developments here. In any case, we calculate this prominence for our function values. And the neat thing of this is, and we will be using that later on in the second part of the talk, is that we can calculate those topological features or those prominence values also in higher dimensions. So it doesn't only require a one-dimensional function, kind of like a mountain range that I showed you, but it also works for higher dimensional functions, for instance, for a 2D function, like this mountain range here that I'm showing you in 3D. And we are always able to derive these prominence values for different peaks and valleys in the data. Again, the details of how this works are not super important at this point, but just know that this process is also known as persistent homology. So there we have this persistence or prominence again, and we obtain as a result a set of topological descriptors, the so-called persistence diagrams. One of them is being shown on the right-hand side. Now, let's end this digression and let's go back to MALDI-TOF and see what we can do with this. Well, once we have our spectrum here, we can calculate these prominence values for every point, and we can just map the spectrum from the intensity domain to the prominence domain or to the persistence domain. And now let's see how that looks for this example. You can see that I used the old spectrum, the unpreprocessed one, in the background, and it's shown in gray here. And you can see that the new one is being shown in black. And one of the perks, one of the results of this transformation is that we get a very nice stable baseline, and we immediately get something akin to a prominence or relevance of our peaks. Notice, for instance, that this guy here, which has a very high intensity in the previous calculation, actually gets assigned a relatively low intensity now because it's part of this smaller range. So essentially, this transformation is not maintaining the original intensity values, but it's mapping them to some other kind of domain, which is a little bit more efficient to deal with. And of course, the nice thing about this is that it doesn't need any parameters at all. It's basically just a prescribed way to say, well, we just map all the peaks in the function to their relevant prominence values, and then we're done. So no parameter tuning necessary for this. Now, how can we use such spectra now for machine learning tasks? Regardless of how they're being calculated, we need some way to deal with them in a machine learning algorithm. There are multiple ways of going about this, and I'm actually gonna present both of them in some sort of fashion. The first one is, let's say, the most straightforward one, or the one that you would do when you do classical machine learning, maybe, and that is you choose a bin size to obtain fixed size feature vectors. So you just bin your data and that's it. The second one is you could use a sparse representation of your spectrum based on some tuples. So you just store the tuple as X and Y coordinates. This latter part is a little bit reminiscent of the deep sets approach, in which you are able to learn from unordered sets of data. Now, what we did is we picked this approach, but we did it in a somewhat different fashion. We defined what we call PIKE, the peak information kernel. And this kernel is a dissimilarity measure that takes two spectra, S and S prime, and calculates the following expression, which might look a little bit horrible at the first glance, but I'm going to show you a picture later on, and you will see what it actually means. Never mind the exact details of this. The PIKE kernel essentially represents a spectrum as a kind of diffusion process, as a kind of heat diffusion of the data, and it represents the X values and the intensity values of the spectrum in order to see how close they are. Now, the nice properties of this kernel are that we're essentially dealing with a C infinity function that has a closed form gradient expression. It might sound a little bit like mumbo jumbo, but what I mean by this is that we can effectively prop gradients through this function because everything here is differentiable. And so we can potentially tune the T parameter, which we'll see in a second, how that goes, and obtain a sort of data-driven way to assess our similarities. Now, overall, the similarity of this kernel is being calculated based on the distances between peaks. So how far are the peaks in terms of their M over C values? And it's also possible, since this goes over all pairwise interactions, we're capable of capturing interactions between peaks. So it's not just something that says, oh, if your peak is at this specific position and you shift a little bit, we are still capable of capturing this. We also only have a single parameter here, namely T, and this controls the smoothing. So how much information we want to keep in the spectrum. And the nice thing about this expression is that we can now easily integrate this into any kernel-based model. So for instance, support vector machine or Gaussian processes or much more. And we actually made use of this in our project in order to obtain a data-driven parameter T that we can easily tune and that we can easily adjust and calculate for different data sets. And we'll see in a few minutes what that actually entails. Now, let me show you an example of what this parameter T does in practice. Varying T is akin to just smoothing or blurring out the full function. So for T almost zero, so no smoothing at all, we just get the original peaks that we supply. If we go a little bit higher, we get something like this. If we go higher, we get something like this, and so on and so forth, until at the end, more or less, we have smoothed out everything and we should get a flat function. Of course, flat functions are not really useful, because if you have the flat function, then everything is similar or almost equal to everything else. So there is clearly a trade-off region, and it makes sense to find the best parameter, find the best T by some process. Now, we picked Gaussian processes for this, and we used a very, very small data set. But as a success story and a small spoiler, I can already tell you that the data set is about to get much, much bigger. But for this first, let's say, pilot project, we used a very small data set consisting of 3 so-called priority pathogens. So this is E. coli, Klebsiella pneumoniae, and Staphylococcus aureus. Those are 3 priority pathogens that have been identified by the World Health Organization to be, well, the most relevant pathogens plaguing most clinics in many countries. We have different antibiotic combinations here. The exact details are not super important, but maybe you've heard of some of them. I hope you didn't have to take them, but some of them are quite well known, such as ceftriaxone or ciprofloxacin. So they're provided, they're given for all kinds of infections in the developed world. The sample sizes, you can see, they are moderate. It's not something that a deep learning person would be very happy about. So for this, we would need a little bit more, but we'll get there. But it works for more classical machine learning approaches. And in particular, it works for approaches like ours, which just take the peaks into account and which use a sparse representation. And this is taken from our paper, Topological and Kernel-Based Microbial Phenotype Prediction from MALDI-TOF Mass Spectra. Now, what we did here is we used a Gaussian process with our PIKE kernel and checked how it performs against a standard logistic regression based on the other pre-processing pipeline that I showed you. So the logistic regression has access to a binned version of the spectrum, so a lot more data, whereas the Gaussian process classifier only gets about 200 peaks that we extracted from the function and provided to this classifier in a sparse fashion. And to our very nice surprise, this performed extremely well, at least in comparison to the logistic regression baseline. We also found that the magic, as it were, or the predictive performance, I should say more formally, the predictive performance actually arises from the choice of kernel. So when we use a different kernel, namely an RBF kernel, for instance, so standard, a classical kernel that doesn't account for interactions of the data, we experience significant performance drops, meaning that the gains that we see for our rightmost section are actually due to the choice of kernel and to the increased expressivity that we gain from that, rather than from picking a better model like a Gaussian process. Now, this is, of course, a classical table that every reviewer likes to see, with everything is bold and so on and so forth. I'm not going to bore you with singing more praises about this, how well this works, because as always, there are advantages and disadvantages. What we found and what we focused on in this paper, since the data set wasn't super big, we were exploiting this capability of doing some sparse processing. And we also used some built-in confidence analysis, because the beauty of the Gaussian process approach is that you get a likelihood for your data. And so you kind of get a calibration almost that makes it possible for our classifier in a specific scenario to reject samples of which it is not sure. Now, this rejection is something that is very critical in the clinic, because if your classifier is, for instance, just trained to detect a specific strain of E. coli, and you give it data from another strain for which it's not trained, then the classifier should raise an alarm and should say, hey, wait a second, I'm not really confident about this. I'm not comfortable processing these types of data because I'm not sure what the prediction would be. So in machine learning, we would, of course, call this distribution shift. And I recently had a very nice discussion with a colleague who said, well, distribution shift might make it sound like it's the data's fault, but it's actually the model's fault. And that is exactly right in our view as well. So we need the model to raise an alarm if it hasn't been trained for this job. Because in the end, if we ever want to use this in the clinical practice and we want to provide patient treatment based on the outcomes of such an algorithm, then we also need to be able to say that something is going on here. So now, enough about this. There is one large elephant in the room that I haven't addressed so far, and that is this model is nice and it has some very nice properties, but fundamentally it suffers from insufficient scalability to larger data sets. The spectra, unless you extract a certain number of peaks, the spectra are increasingly large. You get 20,000 values per spectrum, for instance, because the resolution of the MALDI-TOF MS devices is so good, so high, that you really get a wealth of data. And in some sense, it is a little bit disconcerting or disappointing even that we throw away this data just to gain these results, because surely there must be more hiding in the rest of the data. And to go from this small test data set to a bigger one, we also had to revamp our approach completely. So this is, again, one thing of this zoom and enhance model, where we now zoomed into the data quite a lot and we found, oh, well, it works quite nicely for that. But once we pass over a certain threshold of the number of data points that we have available, we just need a different approach. And this is what we did in our second project, which just came out in Nature Medicine. It's basically a very critical part of Carolyn's PhD thesis, who wrote a splendid thesis and just handed it in at ETH. So I'm really glad that this worked out. And in this data set, we're now finally make every machine learning person's dream come true, I hope, at least when they want to do MALDI-TOF mass spectrometry. So let's see who's up for that challenge. Namely, it's a huge data set of 300,000 mass spectra with almost 800,000 antimicrobial resistance labels. So we have really a battery of different antimicrobials that have been measured, and we have the annotations from the clinicians in there, something like, it's susceptible, it's here it's undefined, or it's indefinite, here it's resistant, and so on and so forth. In total, we have 800 different species of bacterial and fungal pathogens, so it even translates to that, and they're all arising from 4 different diagnostic laboratories. Now, already going to tell you the snack about this. The 4 different diagnostic laboratories, they're all arising from Switzerland, which means that some of the results and certainly some of the distributions of pathogens that we're seeing are, of course, endemic to this region, or maybe endemic to Europe. But I'm already on thin ice here, given my barely existent domain knowledge of this. Nevertheless, it's a start, and I think it's a great start. And the data is also available. So if you want to play with this, if you want to try out your own classifiers, if you want to reproduce our results, go for it. It's available on Dried. I've put the link here. I'll send the slides around. Of course, I was going to say I'm going to put this in the show notes, but it's not a podcast yet. But maybe, who knows? And of course, such a big data set, to get a little bit back to the science here, of course, causes some additional challenges. Namely, we really have to focus on scalability now. So there is no time or no space in this for trying out something like a Gaussian process that uses all types of peak information, even though we like this kernel, and even though I think the kernel has certain properties that make it very nice for such data. We now, for this project, used only something like, I'm using air quotes here, of course, standard classifiers that are based on 6,000 dimensional feature vectors, which we obtained from binning the spectra. So 6,000 arises from the fact that we want to have the M over Z values to be at approximately 5 Dalton, but the exact choice, you could also go for a little bit smaller vectors if your data is incapable of supporting this. But 6,000 worked well for us, and of course it can be easily changed if you want to use more information. The effect of this, and I'm sure that there are smarter ways of solving this, and some people might take us up on this challenge and make this better. We only use this binning in order to obtain fixed size vectors that we could feed into a battery of machine learning models without having to think too deeply about how to process the data in the end. So that in that sense, we were able to train the models and really check the predictions deeply, instead of having to figure out how to provide them with different data sources. Now, let me show you 2 results that are that might be interesting. So first of all, we tried 3 different approaches among lots of other things. So we have quite a lot of data amassed from these for this project in our repositories, which you're also happy to check out, of course, or happy to chat about later. But for now, let's just compare a logistic regression with the light GBM approach. So if you're not familiar with light GBM, it's basically an extension of random forest with some differentiability approaches and improved scalability, and also improved handling of multimodal data or categorical data. Some of those features we used, some of those features we didn't use, but there are certain aspects of light GBM that make it very nice for data analysis, such as the capability of using inbuilt Shapley values. So those are values that can be used for interpretability analysis. So it made for a very nice model here. Anyway, When we take a look at those ROC curves, you can see that the solid line is the logistic regression, the dashed line is the light GBM classifier. In terms of the ROC curves, it seems that for the three priority for this pathogen and the antibiotics, you can see that LR is doing quite well. Light GBM seems to be a little bit worse. When you go to the precision recall curves, you see that this is a little bit different. So the ROC curves are, of course, not the best way to summarize these things, since there is some heavy imbalance going on. Here you can see that in the precision recall curves, the GBM classifier does typically very, very nicely for some of those pathogens, pardon me, for some of those antibiotics, not for others. It's similar in the case of the S. aureus and two of these other antibiotics. So overall, we have now lots of insights into how these classifiers perform, and we can confidently state that in general, the prediction of antimicrobial resistance appears to be feasible with standard machine learning methods. We also have a multi-layer perceptron in there, which also works quite well in some of the cases. But there's not, if I can state this here and go on the record with this, there's not one method that really beats all of them all the time. So it again appears to be kind of a mishmash or a mixed bag, wherein some of the pathogens and some of the antibiotics can be processed with some models in a better fashion than others. And it's our task now, or the task of everyone, of course, who is interested in this topic, to figure out what to do with this, how to stabilize the results, and whether there are some kind of features that can be leveraged to make these predictions more stable and to ensure that almost all the algorithms perform in a similar fashion. But of course, that's not all. So all the results that I showed you before, they are for a single site. So we have, as you might remember from me telling you about when I told you about the data, we have 4 different labs. They differ in size, so not every lab or not every site has the same number of spectra available. There are also, of course, some confounding factors in there, which we'll deal with in a few minutes. But so everything that I showed you has only been done for one site. And one of the things that we wanted to look at in this paper is, of course, the transferability. So what it is, how can you suppose everything works perfectly? You found your model, you found your classifier that gives you perfect antimicrobial resistance predictions. You're willing to roll it out into the clinic, but now your phenotype or your microbial landscape might change over time. Or suppose you open a clinic somewhere else and now you want to roll it out there. It might be possible that the microbial landscape is a little bit different. And so you might want to check how the performance might go here. And this is what we did in a secondary experiment where we took data from one of the sites. So by the way, the data set is called DREAMS for the database of resistance in antimicrobial spectra. And we took one DREAMS site, trained on it, and then we tested on the other DREAMS site. And in an ideal world, almost all the cells here would be kind of the same color. You can see that we're not living in an ideal world here, because when we go from A to B, for instance, we get a little bit better. When we go from A to C, we get a little bit worse, and so on and so forth. So there's clearly some underlying issue here, and we don't get easy domain transferability for these data. Now zooming into this, we of course wanted to figure out what is going on. And this is, I have to say, this is really preliminary work. We tried to submit this to last year's MLHC conference, but it didn't work out, unfortunately. So what we wanted to look at is we wanted to see what is actually the structure of spectra from different sites. Can we say something about this, and can we maybe adjust this here? Can we observe confounding factors? Now, again, our 3 priority pathogens here, E. coli, K. pneumonia, and S. aureus. And now 3 different colors, which I hope you can distinguish. One is for DREAMS B, so for one other side, the green one. The other 2 are from the same site, from the same hospital, but with different mass spectrometry machines. So we have information about which machine was being used in this hospital, because it's a large hospital and they kept excellent records of all the things that they did. And during the time where those spectra were being taken, one machine was put out of order or changed and replaced by a different one. And so there's kind of both machines at place here. Again, in an ideal world, you would be seeing in these dimensionality reduction plots, you would be seeing a mixture of all the points. You wouldn't be seeing direct clusters here. In particular, you should not be seeing a green cluster here, because the green cluster would indicate that it's kind of easy to tell from a spectrum itself whether it has been taken from one of the sites. Now, you can see that this is unfortunately not the case here. So you can see that all these different pathogens have have respectively one very expressed cluster of the second site. So here you have one, here you have another, here you have the third one. So this means that it's quite easy to predict the hospital that was used to do the sample collection just from looking at the spectrum itself. This would make sense if those hospitals were extremely large, were well separated, geographically speaking, because then you would expect a very different microbial landscape. But in this case, they are just separated by about 4 kilometers. So it's really unlikely that there's something going on here. So there's clearly some additional confounding effects at work here that we need to iron out before being able to transfer this directly. It's ongoing, it's preliminary, but we're happy to talk about this and to discuss some approaches. Maybe some of you want to take up this challenge with us. This would of course be amazing. Now, there are some lessons learned for this project here, and then I'm gonna go quickly over the other one. How can I quickly ask, how am I on time? Is it okay if we still discuss the other one?
You have about 4 or 5 minutes left.
Okay, well, maybe then I'll just give you an overview of the other one briefly. Here are some lessons learned for this project. Namely, we were surprised and happy to see that relatively simple models, such as a logistical light GBM and of course a multi-layer perceptron. So we're not talking about deep convolution networks or autoencoders or something like that. We were really surprised that those models already exhibit useful performance. And it turns out that the calibration of classifiers of models is crucial in order to be able to support the rejection of samples. So in order to make an algorithm that you could actually use in the clinic, because you could refuse to process samples that don't fit your trained distribution. We also have some nice additional results in those papers, in particular in the last one, where we analyzed the feature importance to highlight some interesting peaks in a spectrum. So we can actually link that back to peaks that have already been known to drive antimicrobial resistance, because of course that's the second facet of this research that would be fascinating to dive into for the future. Not only figuring out whether it's susceptible or not, but also figuring out, hey, what actually drives this susceptibility or this resistance, of course, right? Now, there's a lot of challenges left here. I only mentioned 2 of them because I felt pessimistic about being able to tackle all of them here. The first challenge is, of course, the domain adaptation. So meaning, how can we find features that generalize to other sites, to other hospitals? But also the second one is, how can we find features that generalize over time? Because there's also a domain shift going on in some sense when you consider the time. The microbial landscape might change, new microbes might appear, new strains might appear, old strains might die out. In some sense, we saw this during the pandemic with the virus, right? We saw that something is still changing, and it's not a static process, and our data need to account for this. And I think that I'm going to stop here now, but I wanted to, of course, acknowledge all my co-authors in the projects that I had the pleasure of working with them. There's quite a lot of people from ETH Zurich, of course, Christian, Karsten, Felix, Michael, Carolyn. It's also our project partners from Basel University. It would be Aline and Adrian. Then there's people from Université de Montréal. That would be Guy Wolf and neuroscientists from Yale University, Smita, Nick, and Tristan. Unfortunately, I didn't have the time to delve into their work too much, but maybe the next time, who knows. The funding for some of those projects was really graciously provided by the Swiss National Science Foundation. And having mentioned MALDI-Quant briefly, so the pre-processing pipeline, I also want to use this to go publicly on record and extend a special thank you to all open source contributors for making our science possible. We also made our source code, of course, publicly available. We would be happy to gain some comments to make it possible for us to collaborate on this. But really, that there's people that invest their time, often their free time, in order to maintain these sort of things. So with that, I'm gonna stop, and thank you very much for your attention, and I'm happily take some questions now.
Thank you, Bastian, for a wonderful talk. Very, very interesting. I believe that we have one question already stated in the chat, and I'll ask Celine to unmute and pose the question.
Hi, Bastian.
Hi.
Nice, like greetings from like Basel, from his previous institute, actually. Really nice. I was also looking at your work with Karstens, like antimicrobial resistance. I was just wondering, like, did you also integrate some other essays, some biochemistry essays or microbiology essays, or some sequencing data that might be interesting for to integrate with the MALDI-TOF data, or maybe using this just as like other types of data to do the same kind of prediction that you have here?
Yeah, that's an excellent question. I have a simple answer for you. Unfortunately, no, we did not yet. So part of this project, or one of the challenges of this project, was to first figure out how to collect all these data. This was a momentous effort from Basel University primarily, and from, of course, the other labs that that provided the data set. We don't have sequencing data available as such for these pathogens, as far as I know. We might have them for some of them, as far as I understand. There are certain sample collections that have been fully sequenced, I think, by Basel University to serve as a test mechanism, but I'm honestly too hazy on the details. I can for sure tell you that we did not put this information into our classifier, at least not for now. That would, I think, be the next frontier for this type of project.
I believe I'll give Celine the opportunity to rebut if she's happy with the answer.
Yeah, I'm happy. Just like I guess, yeah, that will be really helpful, and maybe would help in the sense that sometimes, because you see differences, right, in terms of like strains and also the antibiotics that you applied for these strains, and maybe that could be helpful to better, like, you know, prediction, since you know the background of the strain, maybe genetic.
Oh yeah, yeah. Absolutely. I mean, in some sense, and again, I'm sorry for my lack of biological insight here, but in some sense, so MALDI-TOF is of course not sequencing, right? So the resolution, as it were, from MALDI-TOF is not sufficient to really give you these insights on a cell level. It's more, the interesting thing seems to be that apparently, I mean, judging from the results that we get, MALDI-TOF captures some kind of proxy that tells you something about the resistance mechanism, but to really find the resistance mechanism, it's my understanding that you need to zoom into a little bit more and actually do something like maybe even single cell sequencing.
Yeah. I'm just like, sorry, again, another question, because I'm also working on antimicrobial resistance, but in a different way, trying to understand some certain last resort antibiotics and how they are killing bacteria. So yeah, resistance and understanding the mechanism, I think that could really be helpful for predicting the resistance. I mean, in your case, yeah, and absolutely. Also maybe like NMR data you might be interested to look at. But then again, like I think MALDI-TOF was a good thing because it's, as you said, clinically, you know, at least some of the clinics, maybe in Switzerland, yeah, you can say they have it. Not every hospital might not have NMR, of course, but to maybe not in clinically applied situation, but for for basic science, like trying to understand how resistance develops in a strain. You can use maybe that type of data to figure out, like, because then you can see the interactions with the antibiotic and the strain and what happens there, what changes.
Yeah, absolutely, fully agree. I mean, that's what we're hoping for.
Really nice work. Thanks.
Thank you.
Okay, Bastian, I have another question. So towards the end, you were discussing about this separation that you were seeing in the data sets when you were doing dimensionality reduction and in the different, not class divisions, but basically divisions on where the data was acquired. And you said that you would expect the data to be overlapping. But since you see the overlap in the same hospital, right? So in the blue and the red groups, you see strong overlap, and you don't see an overlap with the green one. Could that be? And I assume that the red and the blue ones, they would be slightly different instruments, right? So they would maybe be the same manufacturer, but maybe slightly different versions of the same. The one was decommissioned and then was updated. Could this difference that you see be due to sample preparation? Like, do you think that this is like that it's just because the different hospitals, they use different ways of preparing the samples before they actually, or it's a different manufacturer altogether that generates things in a different way?
Yeah, I mean, this is
cause for concern because if you don't have a heterogeneous data set from then classification will be heavily dependent on this, right?
Exactly. Yeah. So first of all, let me answer the question in the reverse order, essentially. So first of all, these effects were also the reason why we opted for doing all experiments in the paper that I showed you only on a single site. So essentially, what we do with this paper is we provide the data set as an individual source of MALDI-TOF mass spectrometry, hoping to make it possible to develop new machine algorithms for AMR prediction. We show the labels of the individual sites, and we even make the sites individually available. But for the experiments in the paper, we essentially restricted ourselves to one site because that already had quite some heterogeneous structures in there, for instance, changes over time and so on and so forth. So yeah, we're definitely aware of this, and this is definitely one obstacle to overcome before this might make it into direct clinical practice. Of course, the machine learning answer might be to just retrain on every domain, right? That's certainly a possibility. It's just a little bit inelegant in a sense because you have a cold start problem. You need to acquire much more samples before you're able to roll out this system then. And so ideally it would be possible to do something like a CIFAR 10 or like a trained neural network. You download it and then you say, okay, and now I run it on my, 100 samples that I collected, and now it works quite well for my own domain. So that would be the dream. Now, the sources of these effects, as far as I understand, all the labs were following similar protocols for preparing these samples. So it's not really, it shouldn't really be the case that there are strange or big differences in terms of manufacturers or in terms of how samples were being prepared. But of course, already a little bit the fact that you have some kind of clustering, even with the two machines from the same hospital, maybe that tells us that there's something going on that we don't understand. Maybe there's some step that could be done to remove these type of, let's call them batch effects for now, right? So maybe there's something missing. Maybe we could improve the representations or make them generalized somehow. I think that is going to be a challenge for the future.
Thank you very much, Bastian. I have maybe one comment. Maybe the sample preparation here can learn something from machine learning, like here, like where samples are measured at multiple different sites, and then you compare the outcomes, right? So you use this as a way of training sort of an invariant model to these feature differences or these site-specific differences that we cannot explain. Of course, there will be other problems with this because you cannot have the same sample at the same time at these two different places. But that's a different question, I think. Anyway, thank you very much.
Sorry.
Oh, that was it. I was just thinking that I was just thinking out loud. So yeah, thank you very much for a very interesting talk, and I'm looking forward to following. I'm sure we all are your future work now in Munich.
Thank you. Yeah, I'm looking forward to that as well. So any other questions?
I believe that was all the questions that were. So yeah, thank you, Bastian.
Thank you. Great.